Hello, everyone. My name is Greg Clark, and I am a plant physiologist at the University of Texas at Austin. I want to thank the organizers for inviting me to present my research. The title of my talk today is Altered Expression of Ectoapyrase Enzymes Regulate Both the Level of Extracellular ATP in Growth Zones and the Growth of Primary Roots. I want to start off by introducing you to these enzymes called apyrases. In Arabidopsis, the model plant, there are 7 apyrases, but these enzymes are able to hydrolyze NTP and NDP to NMP. My research is mainly focused on their ability to use ATP as a substrate. Out of the 7 apyrases in Arabidopsis, there are 2, APY1 and AtAPY2, that are 87% identical at the amino acid level. These two apyrases complement each other, and they're both expressed primarily in rapidly growing tissues. They're also both localized in the ECM, where they are hypothesized to function as ectoapyrases, thereby controlling the level of extracellular ATP by hydrolyzing it. So how does ATP get outside of cells? When thinking about extracellular ATP, this is one of the first questions that come to mind. Turns out there are multiple ways that ATP can get outside of cells. Most obvious one is through wounding, when you break open a cell. But there are also transporters, membrane transporters on the plasma membrane that can release ATP. The touch and osmotic stress stimuli can also release ATP. But because plant cells grow by secreting vesicles that include plasma membrane materials, newly synthesized phospholipids and proteins for the plasma membrane, and polysaccharides for the ECM, when a plant cell grows, by definition, it is releasing ATP. So extracellular ATP is a well-known hormone-like signal in animal cells. It's been studied for almost 50 years now, and it induces signaling changes in both animal and plant cells. However, the field of research in plants is a baby of a research field starting about 25 years ago. It's important to note that hydrolysis of extracellular ATP is not needed to activate receptors. In animal cells, there's 2 large families of eATP receptors, P2X and P2Y. However, in plants, the first eATP receptors were discovered a little over 10 years ago, and there are 2 known eATP receptors so far, P2K1 and P2K2. For plant cells, it has also been well documented that some of the earliest signaling changes include an increase in cytoplasmic calcium levels, reactive oxygen species levels, and nitric oxide levels. So one of the early experiments that we did to test the potential role of extracellular ATP in regulating growth was look at treatments with ATP gamma S on the growth of root hairs in Arabidopsis seedlings. So in this data, you see that treatment with 150 micromolar ATP gamma S and 200 micromolar ATP gamma S inhibit the root hair growth rate. We also tested lower levels of extracellular ATP. And in this experiment, you'll see that 15 micromolar ATP gamma S and 25 micromolar ATP gamma S promoted root hair growth in these Arabidopsis seedlings. And this was a very exciting result because this is a characteristic of hormones. One concentration of the hormone does one thing, and another concentration of the hormone does another. So this was one of the first pieces of evidence that extracellular ATP in plant cells, like animal cells, is a hormone-like substance. In this experiment, we tested the effects of extracellular ATP on the growth of primary roots in Arabidopsis seedlings. And what you notice is that we used ATP instead of ATP gamma S. But again, we see this biphasic response in which a low concentration, in this case 137.5 micromolar ATP promoted primary root growth, and a high concentration of ATP inhibited root growth. Interestingly, when we tested the loss of function mutant for the P2K1 plant eATP receptor, we did not see promotion of primary root growth in the 137.5 micromolar ATP treatment. However, we did see inhibition with a higher treatment of ATP. This biphasic response to extracellular ATP has been observed in hypocotyls, pollen tubes, cotton fibers, really every plant cell type and tissue type that we've tested, we've seen this biphasic dose response curve. So if eATP levels regulate primary root growth, so what is the concentration of eATP in growing Arabidopsis primary roots? In order to answer this question, we collaborated with engineers at the University of Florida who designed a microelectrode or a microsensor that could measure extracellular ATP outside of roots of Arabidopsis seedlings. This microelectrode was about 1 micrometer in diameter at the tip, and it worked based on coupling two enzymatic reactions that produced H2O2 that was decomposed at the anode and produced an electrical current that could be measured by an electrode and then converted into extracellular ATP concentration. So when we tested this on Arabidopsis seedlings, we first tested it on wild-type seedlings, and we found that the concentration of extracellular ATP outside of a root is in the nanomolar range. It was a very exciting finding because this would be expected if extracellular ATP is a hormone-like signal. We also found that it varied depending on the zone of the root that we were in. And so about 20 microns from the root tip in the area of the root cap, we saw, which is a highly secretory tissue, we saw higher levels of extracellular ATP, and back in the elongation zone, we saw even higher levels of extracellular ATP. So we had a hypothesis that if eATP regulates root growth and apyrases regulate the level of eATP, then altering the expression of apyrases should alter the rate of root growth. So the double knockout of APY1 and AtAPY2 is lethal because mutant pollen does not germinate. To get around this issue, we made transgenic plants expressing an inducible RNA interference construct that can suppress APY1 expression up to 75% in the background of the APY2 knockout. And in these mutant RNAi lines that are induced by treatment with estradiol, the plants can flower and are fertile, but the adult plants are dwarfs, as you'll see in this next image. So here you're looking at 3 different lines, RNAi lines, and also the single knockout for APY2. And what you observe is that the single knockout has slightly shorter roots, but clearly when you suppress the expression of APY1 in the background of the APY2 knockout in these 3 different RNAi lines, you greatly inhibit root length. Over on this side, you can see the mature plant is a dwarf plant. And in the next slide, what you'll see is quantification of the data you just saw. At 6 days, the APY2 single knockout is statistically significantly shorter root, but at much earlier stages, you have shorter roots in all 3 of the RNAi lines. And the line R24A is the one that has the highest level of APY1 expression suppressed and then up to about 75%. And you can see that its roots are the shortest. So if suppression of apyrase expression inhibits root growth, will suppressing apyrase expression affect ATP, extracellular ATP concentrations? And in doing this experiment, we typically would stain our seedling roots with propidium iodide so we could easily identify the different zones of the root cap area, the meristematic zone, transition zone, and the elongation zone. And what I hope you can see is that in our induced R2/4A RNAi mutant that's suppressed in both expression of both APY1 and APY2, the levels of extracellular ATP are greatly increased compared to the wild type. And here are the quantification for that. So in the R2/4A mutant, you can see at every place that we measured in the wild type, we have a big increase in the concentration of extracellular ATP. So suppression of apyrase expression does increase extracellular ATP in all of these zones. So overexpressing apyrase promotes plant growth, and we were wondering, does this increasing apyrase expression also affect the concentration of extracellular ATP? When we did this experiment, we used two different constitutive overexpressors of apyrases, APY1 overexpressor and APY2 overexpressor. And what I hope you can see is that in all the different zones of the root, the concentration of extracellular ATP is greatly reduced and is consistent in every different place along the root. About half of 1 nanomolar is the concentration. So in my lab, we typically do research first using the model plant Arabidopsis, and then we see if our results can be translated into crops. And so we have this question, does eATP and apyrase expression also affect growth and development of commercially valuable crops? So we took a P ectoapyrase and ectopically expressed it in soybean plants. And one of the first things we noticed is that the root system architecture of these plants grown in the greenhouse was greatly enhanced. And I hope that you can see that in all three of these P ectoapyrase expressing lines, there's improved root system architecture. The seedlings also are typically bigger as well. This improved root system architecture also extended to increase in density and length of root hairs. And this raised the question of whether our transgenic soybean plants might show drought tolerance. So we designed this experiment to test that question. Here you see 26-day-old soybean plants, wild type, and the three transgenic lines. They were grown well watered for 26 days, and then we withheld water for 8 days, and then rewatered these plants for 3 more days. And down here you can see the survival rate. And I hope you can tell that these transgenic lines of soybean their drought tolerance was greatly increased. We wondered whether this expression of the P ectoapyrase might result in increased seed yield. And so under both well-watered conditions and water deficit conditions, we grew our soybean plants to maturity and harvested the seeds. And what we found was that both for the average number of pods per plant and average seed weight per plant, the 3 transgenic lines under both conditions, well-watered and water deficit, had statistically significant increases in the seed yield. These experiments were done in the greenhouse, and experiments done in greenhouse under controlled conditions are often not observed in field-grown plants. So we asked the question, does this apyrase expression affect growth of soybean plants grown in the field? So in multiple years across multiple sites, on the left is data, seed yield data from fields in the Midwest of the US, and on the right is data from fields in Puerto Rico. And what you can see is that in 2 of the soybean lines in the Midwest, there was a statistically significant increase in the seed yield. In all 3 lines in the Puerto Rico field trial, there was statistically significant increase in seed yield. Now, these experiments were done in an older strain called Williams 82, which is no longer used in agriculture. So we wondered whether this increase in seed yield might persist for elite germplasm of soybean plants. So here's the data in patent-protected elite germplasm, and we saw statistically significant increase in seed yield in multiple field trials in multiple sites, and so we're very excited to find that this trait of increased seed yield persists even in elite germplasm. So in summary, what I hope is the takeaway from my presentation is that extracellular ATP and ectoapyrases regulate growth of a variety of plant cells and tissues, that the extracellular ATP levels in Arabidopsis primary roots is nanomolar and is highest in the elongation zone, that the ectoapyrases in Arabidopsis, APY1 and AtAPY2, their expression regulates the level of extracellular ATP in Arabidopsis primary roots. And lastly, that ectopic expression of a apyrase in soybeans increases growth, drought tolerance, and yield. I want to thank the sources of my funding, the Freshman Research Initiative, National Science Foundation, and Texas Crop Science. Also want to recognize my collaborators. Stanley Rue is a co-PI of the lab with me. Eric McLemore and Diana Venegas are the engineers who designed the microsensor, microelectrode that we use to measure extracellular ATP levels. And then Catherine Brown and Ashley Cannon made important contributions to this study, as well as 2 undergraduates, Miranda Jankovic and Ryan Wang. Thank you so much for your attention. I would love to have questions and ideas. Because this is a virtual presentation, I'm putting my email address in case that might be an easier way for you to reach out with questions and ideas.